Good morning, everyone. I'm Dr. Abhishek Pathre. I'm assistant professor in Department of Pathology, Santosh Medical College and Hospital. And today I'm going to take your lecture on the topic is etiopathogenesis of disease, cell injury, and degeneration. So, if we talk about the introduction of these topics, the normal cell is confined to a fairly narrow range of function and structure by its state of metabolism, differentiation, and specialization by constraints of neighboring cells and by the availability of the metabolic substrates. Nevertheless, it is able to handle physiological demands, maintaining a steady state called homeostasis. Adaptations are reversible functional and structural responses to more severe physiological stresses and in some pathological stimuli, during which new but altered steady states are achieved, allowing the cell to survive and continue its function. When the stress is eliminated, the cell can recover to its original state without having suffered any harmful consequences. If the limits of adaptive responses are exceeded, a sequence of events follow that is called as cell injury. Please focus on the word that is cell injury. The cell injury can be reversible up to a certain point, but if the stimulus persists or is severe enough from the beginning, the cell suffers irreversible injury and ultimately cell death can happen. Cell death, which is the end result of progressive cell injury, a normal or essential process is embryogenesis, as we all know, the development of organs and maintenance of homeostasis. There are two principal pathways of cell death, one is necrosis and one is apoptosis. The stresses of different types may induce changes in the cells and tissues other than the typical adaptations, the cell injury and death, in the form of intracellular accumulations and pathological calcifications. And finally, the normal process of aging itself is accompanied by characteristic morphologic and functional changes in the cells. So, if we talk about a normal flowchart of a cell injury, if the injury is mild, then becomes an irreversible injury, and if the injury is moderate to severe, that becomes irreversible injury and ultimately can lead to necrosis or apoptosis. Sometimes the injury is after injury the cell is able to adapt. So, if we talk about the stresses which can cause the cell injury, which can be divided into many parts, the first is the physical, which can be mechanical trauma, thermal, pressure, radiation, waveforms as well as electricity. The chemical one, which can be organic as well as inorganic. The microbial, which can be bacterial, viral, fungal, parasitic, and so on. Immunological includes innate and adaptive immunity. Nutrition includes undernutrition as well as overnutrition, and genetic, that is inherited or sporadic. The cell adaptations include four major points. The first one is hypertrophy, atrophy, hyperplasia, and metaplasia. We are going to discuss all these points in details. So, the first one is hypertrophy. Hypertrophy refers to an increase in size of the cells or organelles resulting in increase in the size of the organ. Please focus. The hypertrophy means increase in the size of the cells, not increase in the size of the number. So, which is very important. In hypertrophy, there is no increase in the number of cells, only the size of the cells are increased. The hypertrophic organs has no new cells, very important. Occurs in organs and tissues where the predominant cell population is in G0 phase of the cycle. The increased size of the cells is due to synthesis of more structural component of the cells. Now, the second is hyperplasia. Now, in hyperplasia, there is increase in the number of cells. Please focus. In hypertrophy, there is increase in the size of the cells, whereas in hyperplasia, there is increase in the number of cells in an organ or tissue, usually resulting in increased mass of organ or tissue. Now, the third is atrophy. First was hypertrophy, then hyperplasia. Now, the term is atrophy. Atrophy refers to a decrease in the size of the cells or cell organelles, resulting in decrease in the size of the organ. And finally comes the metaplasia. Metaplasia is a reversible change. Please focus the term reversible. Metaplasia is a reversible change in which one differentiated cell type, that can be epithelial or mesenchymal, is replaced by another cell type. Okay, which is a very important term. If we talk about the malignancies or the neoplastic changes, the initial change is metaplasia, which is a reversible. So the basic principles in the cell injury include the cellular response to injurious stimuli depends upon the nature of the injury, its duration and its severity. The consequences of the cell injury depend on the type, the state, the adaptability of the cell. The cell injury results on different biochemical mechanisms acting on several essential cellular components. Any injurious stimulus may simultaneously trigger multiple interconnected mechanisms that damage the cells. Mechanisms of cell injury includes depletion of ATP, mitochondrial damage, loss of calcium homeostasis, oxidative stress, altered membrane permeability, and endoplasmic reticulum stress. The first is depletion of ATP. The causes can be reduced supply or impaired utilization of the oxygen or the nutrients. Mitochondrial damage. Mitochondrial damage can be after hypoxia, toxins, or mutations in the mitochondrial genes, which are the causes of some inherited disorders. Now, the significance of mitochondrial damage is key. This is a very important structure in our cell, and damage can lead to decrease in the homeostasis. The loss of homeostasis can also be due to ischemia and toxins. The oxidative stress, which is the inability of the free radical scavenging system. Now, this free radical term is very important these days, and all the products which is we are eating can have the free radical that can damage our cells. So, we should have those products that inhibit the production of these free radicals, because these free radicals, these are not, if we not clear the reactive intermediates during the cell metabolism, thereby resulting in an excess of free radicals. The causes can be chemical injury, radiation injury, ischemia reperfusion injury, cellular aging, inherited defects, and microbial killings by phagocytes. Classification of the free radicals include electron transport chain, lipid peroxidation, phagocytes, and nitric oxide. Nitric oxide, a molecule generated in many tissues, is a potent vasodilator and mediator of a number of important biological processes. The defects in membrane permeability. The integrity of the cell is lost when there is a defect in membrane permeability. The causes can be ischemia, bacterial toxins, viral proteins, lytic component systems, numerous physical and chemical agents. So, the death of a cell or the injury to a cell can happen in many ways: when there is decrease in the ATP, when there is mitochondrial damage, when there is altered calcium entry into the cell or exit from the cell, when there is increased reactive oxygen species, when there is membrane damage, and when there is protein misfolding and DNA damage. So, when the injury is reversible, cell is able to regain its function back to a normal state. When the injury becomes irreversible, initially the biochemical alterations happens, then ultrastructural changes occurs, then lead to light microscopic changes, means the changes we can see under the light microscope, and finally the gross morphological changes occurs and cell death can happen. Reversible injury features include cellular swelling, hydropic change or vacuolar degeneration, increased eosinophilia of cytoplasm, and fatty change. Features of reversible cell injury can be recognized under electron microscope in the form of plasma membrane alterations like blebbing, blunting, loss of microvilli, mitochondrial changes like swelling and the appearance of small amorphic densities. ER changes include dilatation of endoplasmic reticulum with detachment of the polysomes. Intracytoplasmic myelin figures may also be seen, and nuclear alterations in the form of disaggregation of granular and fibrillar elements. Apoptosis, a very common term we all read in pathology, and not in pathology, after the pathology also, because apoptosis is an integral part of human homeostasis. Without apoptosis, the chances of cancer, because it is programmed cell death, and if the programming of cell death is not good, that can lead to cancer or neoplasia. So if we define apoptosis, it's a programmed or pre-arranged pathways of cell death triggered by a variety of extracellular and intracellular signals in the form of absence of inflammation. Please focus. There is, if we focus on the key words of apoptosis, the first is it is a programmed cell death and there is absence of inflammation. It is recognized in 1972 by the distinctive morphological appearance of the membrane bound fragment derived from the cells and named after the Greek designation called filing off. The mechanism of apoptosis can be divided into two phases. The first one is the initiation phase, where there is activation of the caspases happens, and the second one is the execution phase, where degradation of the cellular components occurs. The initiation phase is further divided into two parts or the pathways. The first one is the intrinsic or the mitochondrial pathway, and the second one is the extrinsic or the death receptor initiated pathway. So the first question which occurs into our mind is, what are caspases? Caspases, these are cysteine proteases. Unique ability of these enzymes to cleave after aspartic acid residues. Caspase families include more than 10 members. The presence of cleaved active caspases is a marker for cells undergoing apoptosis. Initiation में caspases, the initiation phase, the caspases which are involved are caspases 8 and caspase 9. Please remember and focus on these caspases as these are asked in many questions and exams. Execution caspases are 3 and 6. As we all know, the intrinsic pathway or the initiation phase is divided into two parts. The first one is the intrinsic or the mitochondrial pathway, major mechanism of apoptosis in all the mammalian cells. Results of increased mitochondrial permeability and release of pro-apoptotic molecules into the cytoplasm. The mitochondrial proteins include BCL family, the anti-apoptotic caspases or this mitochondrial protein. These are BCL2, BCL6 and MCL1. The pro-apoptotic are BIX, BIM and BAD. So how do these caspases get activated? The next question is, the two hypotheses that have been provided for the activation of caspases are, the first of these was an allosteric model in which the monomer is activated directly by the apoptosome. Okay. In the second, the induced proximity model, the apoptosome provides a platform for caspase 9 dimerization. Now the clearing of apoptotic cells. The plasma cell membrane of apoptotic cells damage in ways that promote the recognition of the dead cells by phagocytes. One of these changes is the movement of some phospholipids, notably phosphatidylserine, from inner leaflet to the outer leaflet of the membrane, where they are recognized by a number of receptors on phagocytes. These lipids are also detectable by binding of a protein called annexin V. Some of the apoptotic express thrombospondin, an adhesive glycoprotein that is recognized by phagocytes and macrophages themselves, and may produce proteins that bind to apoptotic cells, and thus the targets dead cells for engulfment. Apoptotic bodies may also coated with natural antibodies and proteins of the complement system, notably C1q, which are recognized by the phagocytes. Examples of apoptosis are programmed destruction of dead cells, involution of the hormone dependent tissues, death of host cells that have served their usual purpose, DNA damage, accumulation of misfolded proteins, cell death in certain infection, pathological atrophy in parenchymal organs after obstruction. Light microscopic changes include round or oval mass of intensely eosinophilic cytoplasm with fragments of densely nuclear chromatin, absence of inflammation. Electron microscopy shows cell shrinkage, chromatin condensation, formation of cytoplasmic blebs and apoptotic bodies, phagocytosis of apoptotic cells or cell bodies, usually by macrophages. Now the current focus on research in apoptosis are bio-oncology, neurodegenerative disorders and immunological disorders. Targeted therapies are also included, which is a part of research analysis. So the main focus of this is apoptosis and necrosis. The examples of apoptosis we all know during embryogenesis, during menstruation cycle, when there is like formation and death of the tissues happens, during formation of any regeneration of any organs, and during involution of the thymus. All this is like part of apoptosis. Whereas in case of necrosis, which usually happens after, when there is any infection or inflammation which is going on, which means okay, some inflammation or infection is going on causing death of the cell. So which is very important during this. Okay, so the today's topic we have discussed major parts of cell injury, cell regeneration. Apart from this we have discussed about apoptosis and necrosis. And the thing is, the important terms, if we talk about the important terms which we come usually during our teaching, is one is hyperplasia, then other is hypertrophy, then comes atrophy, metaplasia, dysplasia, and necrosis and apoptosis. So this is all for today. I hope you guys understand the major parts and topics and the definitions of all these terms, which is very important, because when we move forward and read further about pathology, these topics and terms will frequently come. So have to focus about all these. Thank, thank you everyone. Thank you. If you want to ask any question, you can.